There is something deeply unsettling about what happened on Mars in the summer of 1976. Two identical spacecraft touched down on the surface of the red planet, 4,000 miles apart. They carried robotic arms, cameras, and a suite of sophisticated instruments designed to answer one of the most profound questions in human history. Is there life on Mars? And for a brief moment, the answer appeared to be yes. The Viking landers detected something in the Martian soil that consumed nutrients, released radioactive gas, and behaved exactly like living microorganisms. For most scientists, this was proof that the readings were false. But nearly 50 years later, a different interpretation has emerged, one that is far more disturbing. What if Viking did find life on Mars? And what if, in our attempt to detect it, we accidentally killed it? This is the story of the most controversial biological test ever conducted on another planet. Let's start at the beginning. August 20, 1975, NASA launches Viking 1 from Cape Canaveral. 3 weeks later, Viking 2 follows. Each spacecraft consists of two parts: an orbiter to photograph Mars from above and serve as a communication relay, and a lander to touch down on the surface and conduct experiments. The journey takes nearly a year. On July 20, 1976, Viking 1 settles onto the rocky plains of Cris Planitia, 23 degrees north of the Martian equator. 6 weeks later, Viking 2 lands at Utopia Planitia, 48 degrees north. They are separated by 4,000 miles. Both landers carry the same suite of instruments, 4 biological experiments designed to search for life. The first is called the gas exchange experiment. It exposes Martian soil to humidity, then to water, then to a complex mixture of organic nutrients. Scientists monitor for gases that might be released by metabolizing organisms. The second is the pyrolytic release experiment. It tests whether Martian microbes might use photosynthesis to incorporate carbon from the atmosphere into biomass. The third is the gas chromatograph mass spectrometer. Not a life detection experiment, exactly, but critical nonetheless. If life exists on Mars, there should be organic molecules in the soil. The instrument is sensitive enough to detect them at parts per billion. And the fourth is the labeled release experiment. This one gives the most promise. The concept is elegant, simple, and extremely sensitive. A small sample of Martian soil is placed in a sealed test chamber. Then, a nutrient solution is added, just a few drops, 0.115 milliliters to be exact. The nutrients are compounds that exist on Earth. Simple organic molecules like formate, lactate, glycine, and alanine, the kind of molecules that could have formed naturally on early Mars through the same processes that created them on early Earth. But here's the key. Every carbon atom in these nutrients is tagged with carbon-14, a radioactive isotope. If microorganisms are present in the soil, they should consume the nutrients as food, and when they metabolize them, they should release carbon dioxide. Radioactive carbon dioxide. The air above the soil is continuously monitored. Any radioactive gas that appears is counted by a detector. If the counts rise, it means something in the soil is eating the food. This is a metabolic test. It doesn't depend on culturing organisms in a lab. It doesn't require them to grow or reproduce. It simply detects their activity in real time. The method is so sensitive that laboratory tests on Earth detected as few as 30 bacterial cells in a sample. Now, the critical part. Every positive response must be verified with a control. A duplicate sample of the same soil is heated to 160 degrees Celsius for 3 hours. This temperature is hot enough to kill any known microorganism on Earth. If the active response is biological, the heated control should show no response. If the response is chemical, heating shouldn't change anything. That's the test. That's how you distinguish life from chemistry. July 30, 1976. Sol 10 of the Viking 1 mission. The first labeled release test begins. The soil sample was collected 2 sols earlier by the lander's robotic arm, dredged from about 4 centimeters deep. It's been stored in the sample distribution box at roughly 10 degrees Celsius. The sample is loaded into the test chamber. The chamber is sealed, the nutrient solution is injected, and immediately, the radioactive gas count begins to rise. Not gradually, not after a delay, immediately. The response is strong, within the range of what you would expect from viable soil on Earth. The gas continues to evolve for about 2 sols, then begins to level off. By sol 7, the curve has nearly plateaued. At this point, a second injection of nutrient is added. If living organisms consumed the first batch of food and are still alive, they should respond to more. The counts should rise again, but they don't. Instead, the counts drop. The radioactive gas in the chamber decreases by about 25%. Then it slowly begins to re-evolve. This is strange, but it's not necessarily bad news for biology. The reabsorption of gas is consistent with a physical-chemical reaction involving carbon dioxide, water, and the alkaline Martian soil. Laboratory tests later confirmed this could happen even without organisms present. The important result is the initial response. The strong, immediate evolution of radioactive gas following the first nutrient injection. Now comes the control. A duplicate sample of the same soil is heated to 160 degrees Celsius for 3 hours. Then it's injected with the nutrient solution. No response. The heated soil is dead. Taken together, the active response and the negative control are exactly what you would expect from a biological system. Scientific protocol demands replication. So a third test is conducted. A fresh sample from the same area. A new active sequence. Once again, the response is positive. Strong radioactive gas evolution immediately following nutrient injection. The result from the first cycle is validated, and then something unexpected happens. The fourth and final test at Viking Site 1 uses soil that has been stored in the sample distribution box for 141 sols, nearly 5 months. The sample has been kept in the dark, open to the Martian atmosphere, at temperatures ranging between 10 and 26 degrees Celsius. 2 nutrient injections are made, 3 hours apart. Both produce null responses. No radioactive gas. The soil is inactive. Long-term storage at these temperatures has inactivated whatever was producing the signal. For a chemical oxidant, this makes no sense. Chemicals don't die from sitting in the dark for a few months. But for living organisms adapted to the extreme cold of Mars, prolonged exposure to 10 to 26 degrees Celsius could be lethal. They would be outside their normal environment, stressed, eventually killed. This looks like biology. Now let's go to Utopia Planitia, 4,000 miles away. The first labeled release test at Viking Site 2 produces the same result. Strong positive response. Immediate evolution of radioactive gas following nutrient injection. It's essentially a duplicate of what Viking 1 found. But now the Viking biology team wants to try something different. Some scientists have questioned whether heating soil to 160 degrees is really proof of biology. Maybe the heat is affecting the chemistry in ways we don't understand. So, they agree to test a lower temperature, 50 degrees Celsius. This is well below the boiling point of water. Most chemical oxidants wouldn't be affected at all, but for Martian microorganisms acclimated to temperatures far below freezing, 50 degrees could be harmful. A fresh sample is collected. The booster heater in the test chamber is activated. The soil reaches 51 degrees Celsius and is held there for 3 hours. Then it's allowed to cool. Then the nutrient is injected. The response is drastically reduced. Only about 15% of the gas that was produced in the first cycle, and the kinetics are strange. Instead of a smooth curve, there are several small peaks, something the engineers have never seen before. They check the instrument. No anomalies. It's functioning normally. So they run a fourth test to clarify what happened. Another fresh sample. Another low-temperature pre-treatment. This time the soil reaches 46 degrees Celsius. The response is stronger than the 51-degree test, but still reduced. About 30% of the gas produced in the first cycle. The conclusion is clear. Preheating the soil to around 50 degrees significantly reduces the activity of whatever is producing the signal, and the degree of reduction appears to be temperature dependent. Few chemical agents would show this kind of sensitivity to such low temperatures, but living organisms would. There's one more variable to eliminate. Some scientists suggest that the positive responses could be caused by ultraviolet radiation from the sun. Mars has no ozone layer, so the surface is bathed in intense UV light. Maybe the UV is creating reactive oxidants in the soil, and those oxidants are what's reacting with the nutrients. To test this, the sampling arm moves a rock. The rock is called notch rock. It's been sitting on the surface for eons, shielding the soil beneath it from sunlight. A sample is collected from under the rock. It's placed in the test chamber and left for 2 sols. Then the nutrient is injected. Strong positive response. The UV theory is dispelled. Whatever is producing the signal, it exists even in soil that has been protected from sunlight for geological timescales. And there's one final test at Viking Site 2. The same long-term storage experiment conducted at Viking Site 1. Soil is collected and stored in the sample distribution box for 84 sols at approximately 10 degrees Celsius. Then it's tested. No response. The soil is dead. Once again, long-term storage at relatively warm temperatures has completely inactivated the sample. Now we need to talk about what went wrong. Because despite the positive results from the labeled release experiment, the scientific community did not conclude that the Viking had found life. The reason is simple. The gas chromatograph mass spectrometer found no organic molecules in the Martian soil. None. This was shocking. Organic compounds are common in the universe. They're found on asteroids, meteorites, comets. They rain down on planetary surfaces constantly. Even if Mars never had life, scientists expected to find organics from meteorites. But the instrument detected nothing. Martian soil appeared to contain less carbon than the lifeless lunar soil returned by the Apollo missions. This was the decisive factor. If there are no organics on Mars, then there can't be organisms consuming organics. The labeled release results must have another explanation. For 30 years, that was the consensus. But then, something changed. August 2008, NASA's Phoenix lander touches down near the North Pole of Mars. One of its instruments analyzes the chemistry of the soil, and it finds something unexpected. Perchlorate. Perchlorate is a salt, a chlorine atom surrounded by 4 oxygen atoms. It's a powerful oxidizer when heated, but at low temperatures it's relatively stable. On Earth, it's used in fireworks and rocket fuel. Phoenix detects perchlorate at concentrations of about 0.4 to 0.6% by weight. This is significant because perchlorate does something very specific when it's heated above 200 degrees Celsius. It releases oxygen, and in the presence of organic molecules, it oxidizes them, burns them into carbon dioxide. The gas chromatograph mass spectrometer on Viking worked by heating soil samples to high temperatures, 200 degrees, 350 degrees, 500 degrees. The gases released were then analyzed. If perchlorate was present in the Viking samples, it would have destroyed any organics during the heating process. The very method used to search for organics would have obliterated them. Suddenly, the absence of organics isn't proof that they never existed. It's just proof that the experiment couldn't detect them. In December 2010, a team of scientists publishes a paper in the Journal of Geophysical Research. The lead author is Rafael Navarro Gonzalez from the National Autonomous University of Mexico. The co-author is Chris McKay from NASA Ames Research Center. They conduct a simple experiment. They take soil from the Atacama Desert in Chile. This is one of the driest places on Earth, and it contains Mars-like levels of organic carbon, about 32 parts per million. They add 1% magnesium perchlorate to the soil. Then they heat it using the Viking protocol. The result is striking. Nearly all the organics are destroyed, oxidized into water and carbon dioxide, but a small amount is chlorinated, converted into chloromethane and dichloromethane. These are the exact compounds that Viking detected. At the time, Viking scientists interpreted them as contaminants, cleaning fluids from Earth that had contaminated the instruments during assembly. They weren't considered indigenous to Mars. But now there's another explanation. They could be the byproducts of Martian organics reacting with perchlorate during the heating process. The researchers run the numbers. Based on the amount of chloromethane and dichloromethane detected by Viking, they estimate the Martian soil could have contained between 0.7 and 6.5 parts per million of organic carbon. This is a small amount, but it's not zero, and it changes everything. There's one more piece to this puzzle. When Viking detected chloromethane and dichloromethane, the scientists measured the ratio of two chlorine isotopes, chlorine-35 and chlorine-37. On Earth, this ratio is about 3:1. The Viking measurements matched the Earth ratio, which is why the compounds were interpreted as terrestrial contaminants. But here's the problem. At the time, no one knew what the chlorine isotope ratio was on Mars. If Mars had a very different ratio, that would be strong evidence that the chlorinated compounds were contaminants from Earth. But if Mars had the same ratio as Earth, it wouldn't tell us anything. In 2021, the Trace Gas Orbiter measures the chlorine isotope ratio in the Martian atmosphere. It's almost indistinguishable from Earth's. The isotope data is inconclusive. It doesn't prove the compounds are Martian, but it doesn't prove they're terrestrial either. There's still the question of what caused the labeled release results. In 2013, astrobiologist Richard Quinn at NASA Ames conducts a series of experiments. He shows that when perchlorate is exposed to gamma radiation, it produces hypochlorite. Hypochlorite is a strong oxidizer, and when it's mixed with amino acids, like the ones in the Viking nutrient solution, it rapidly oxidizes them into carbon dioxide. Radioactive carbon dioxide. This could explain the Viking results without invoking biology. But there are problems with this hypothesis. First, hypochlorite should be stable at 50 degrees Celsius. It shouldn't be significantly affected by the low-temperature sterilization tests that Viking conducted. But the Viking data clearly shows a strong temperature dependence. Second, hypochlorite should be destroyed by long-term storage in the dark at 10 degrees Celsius. Wait, actually, it shouldn't. Chemical oxidants don't die from sitting in a box for a few months. But the Viking data shows that long-term storage completely inactivated the soil. Quinn's team acknowledges these limitations. They state that further experiments are needed to characterize the thermal stability of hypochlorite and other oxychlorine species in the context of the labeled release experiment. As of now, those experiments have not been completed. Now we come to the most provocative interpretation. In 2007, two scientists publish a paper, Dirk Schulze-Markuch from the Technical University Berlin and Joop Hout-Cooper. They propose something radical. What if the Viking experiments didn't fail to find life? What if they found it and then killed it? The key is water. The labeled release experiment was designed under the assumption that all life requires liquid water, so the nutrient solution contained water. 0.115 milliliters of it dropped onto half a cubic centimeter of soil. For most life on Earth, this makes sense. Water is the universal solvent, the medium in which biochemistry occurs. But Mars is not Earth. Mars is hyper-arid. The driest desert on our planet is the Atacama in Chile. It receives less than 15 millimeters of rain per year in some regions. And even there, microbial life exists. But it doesn't exist in the wet parts. It exists in salt rocks. Schulze-Markuch has studied these microbes extensively. They survive by extracting water from the atmosphere using hygroscopic salts, salts that attract and absorb moisture from the air. These organisms can tolerate extremely low water activity, but they cannot tolerate flooding. In fact, when torrential rains hit the Atacama Desert in 2017, scientists found that 70 to 80% of the indigenous bacteria died. They literally drowned. Too much water, too fast. Now imagine Martian microbes. adapted to an environment far drier than the Atacama, surviving for billions of years by extracting trace moisture from hygroscopic salts in the soil. Then a robotic lander from Earth adds liquid water. They drown. Schulze-Makuch goes further. In a 2007 study, he and Houtcooper proposed that Martian microbes might not use water as their intracellular fluid. They might use a mixture of water and hydrogen peroxide. This sounds insane. Hydrogen peroxide is toxic to most life on Earth. It's an oxidizer. It damages cells, but it has advantages in the Martian environment. First, it lowers the freezing point. A 30% solution of hydrogen peroxide in water freezes at around -40 degrees Celsius. This would allow liquid to exist inside cells even in the extreme cold of Mars. Second, it provides a source of oxygen. Microbes could decompose the hydrogen peroxide to release oxygen for metabolism. Third, it's hygroscopic. It absorbs moisture from the atmosphere. Now, if this hypothesis is correct, the Viking experiments would have been catastrophic for Martian life. The labeled release experiment added water. This would dilute the hydrogen peroxide inside the cells, disrupting their chemistry. It might kill them. The gas chromatograph mass spectrometer heated the soil. If the cells contained hydrogen peroxide, heating would cause it to react violently with any organic molecules in the vicinity. This would produce large amounts of carbon dioxide, which is exactly what the instrument detected. The gas exchange experiment added even more water, a complex nutrient broth. This would flood any hydrogen peroxide-based cells, destroying them completely. Every experiment designed to detect life would instead kill it. But wait, if Mars is so dry, why would we assume life needs water at all? Because Mars wasn't always dry. Billions of years ago, Mars had rivers, lakes, possibly oceans. The surface was warm enough for liquid water to exist. Then something changed. The planet lost its magnetic field. Its atmosphere was stripped away by solar wind. The surface became cold and arid. But if life emerged during the wet period, it would have had billions of years to adapt, to evolve strategies for surviving in an increasingly dry world. And recent missions have found evidence that water still exists on Mars. In 2008, the Phoenix lander photographed frost at its landing site. Water ice, condensed from the atmosphere during the Martian night. In 2015, NASA announced the discovery of recurring slope lineae, dark streaks that appear on Martian hillsides during warm seasons and fade during cold seasons. Spectroscopic analysis suggests they might be caused by briny water, kept liquid by dissolved salts like perchlorate. In 2018, the Mars Express orbiter detected what appears to be a subsurface lake beneath the South Polar ice cap. Radar data suggests liquid water trapped under more than a kilometer of ice. Water is not absent from Mars, it's just hidden, locked in ice, absorbed in salts, buried underground. For microbes living in the soil, hygroscopic salts could provide enough moisture to survive. Deliquescence, the process where salts absorb water vapor and dissolve into brine, could create microscopic pockets of liquid, even in the hyper-arid surface environment. This is how life survives in the Atacama. And if life exists on Mars, this is likely how it survives there too. There's one more piece of evidence worth considering. In 2009, scientists using ground-based telescopes on Earth detected methane in the Martian atmosphere. Not everywhere, but in specific locations, and not all the time. The methane appeared seasonally, then disappeared. Methane on Mars is puzzling because it shouldn't last long. Ultraviolet radiation from the sun breaks it down in a few hundred years. If methane is present, something must be replenishing it. There are two main possibilities: geological activity, like volcanic outgassing or reactions between water and rock, or biological activity, methanogens, microorganisms that produce methane as a metabolic byproduct. The Curiosity rover has also detected methane, but only at night. During the day, it disappears. This suggests the methane is being released from the subsurface and then quickly destroyed by some chemical process in the atmosphere or soil. One hypothesis, proposed by Gilbert Levin, the principal investigator of the Viking labeled release experiment, is that Mars might have an active methane cycle. Methanogens producing methane underground, methanotrophs consuming it at the surface. This would explain both the seasonal appearance of methane and its rapid disappearance. It would also suggest that Mars has a functioning biosphere. Let's summarize what we know. The Viking labeled release experiment produced positive results at both landing sites. The responses were similar in magnitude and kinetics to results from viable terrestrial soils. The responses were eliminated by heating the soil to 160 degrees Celsius for 3 hours. This is consistent with the destruction of microorganisms. The responses were significantly reduced by heating to approximately 50 degrees Celsius. This temperature sensitivity is unusual for chemical reactions but expected for biological systems. The responses were eliminated by storing the soil in the dark at 10 degrees Celsius for several months. Chemicals don't die from storage, organisms do. The responses occurred even in soil that had been shielded from ultraviolet light for eons. The signal is not an artifact of UV activation. The responses were widespread. Viking 1 and Viking 2 were 4,000 miles apart, yet both produced similar results. When a second injection of nutrient was added to active soil, about 25% of the gas was reabsorbed. This is consistent with physical-chemical reactions involving carbon dioxide and alkaline soil. But the fact that no additional gas was produced suggests the active agent was no longer present. It had been consumed, or it had died. All of these characteristics are consistent with biology. Now let's look at the case for chemistry. The gas chromatograph mass spectrometer found no organic molecules in the Martian soil. If there are organisms consuming organics, there should be organics present. But we now know that perchlorate could have destroyed any organics during the heating process, so the absence of organics is not conclusive. The low-temperature sterilization at 50 degrees should have destroyed hypochlorite, produced by irradiated perchlorate. This could explain the reduced response. But hypochlorite should not be destroyed by long-term storage at 10 degrees, so the storage tests remain unexplained by the hypochlorite hypothesis. Superoxides, created by ultraviolet light, reacting with carbon dioxide and soil, could oxidize the nutrients to produce radioactive gas. But superoxides should be present in all Martian soil, including soil shielded from UV light. And the Viking data shows that shielded soil still produced a positive response. So UV activation is not required. No chemical hypothesis has yet explained all of the Viking data. In 2016, Gilbert Levin and Patricia Ann Straat, the investigators who designed and operated the labeled release experiment, published a comprehensive review in the journal Astrobiology. Their conclusion is direct. No non-biological explanation as of that date is able to satisfactorily account for all of the Viking labeled release data. Specifically, no chemical hypothesis addresses the question of what active agent in the soil could be adversely affected by heating to approximately 50 degrees Celsius and destroyed by long-term storage in the dark at 10 degrees Celsius. These thermal characteristics are strongly suggestive of biology. Levin and Straat state that biology should be maintained as a viable explanation for the labeled release results, not because it's proven, but because it has not been disproven. And given the possibility that astronauts will eventually explore Mars, biology should be held in the forefront of possible explanations. For the health and safety of those astronauts, there's one more complication. Viking was the only Mars mission that underwent rigorous heat sterilization before launch. The landers and their instruments were baked to reduce microbial counts. No other Mars mission has been sterilized to the same degree. The Mars Pathfinder, Spirit, Opportunity, Curiosity, and Perseverance rovers were not heat sterilized. They were cleaned and assembled in controlled environments, but they were not sterile. This means that every spacecraft we've sent to Mars since Viking could have carried terrestrial microbes to the surface. If those microbes survived, they could have contaminated the Martian environment. And if Martian life exists, It could now be competing with Earth life, hybridizing with it, or being driven to extinction by it. This is why the Viking labeled release data is unique. It's the only data we will ever have on biologically pristine Martian samples. We can never repeat the experiment under the same conditions. So what now? Dirk Schulze-Makuch argues that future missions should adopt a new strategy. Instead of following the water, we should follow the salts. Specifically, hygroscopic salts, sodium chloride, potassium chloride, chlorates, perchlorates. These salts can pull moisture from the atmosphere. They create microenvironments where water activity is high enough for microbial life, even in a hyperarid desert. On Mars, regions rich in these salts could be oases for life. The southern highlands of Mars show high concentrations of chloride. This makes it a prime target. Schulze-Makuch also argues for a different approach to life detection. Don't rely on one experiment. Use multiple independent methods. Microscopy. Look directly for cells. Motility tests. Track the movement of particles in a drop of liquid. Microbes move in characteristic patterns. Sediment particles move differently. With artificial intelligence, we could distinguish between them. Gas chromatography and mass spectrometry. But use lower heating temperatures or chemical extraction methods that don't destroy organics. Chiral tests. All known life on Earth preferentially uses left-handed amino acids and right-handed sugars. Chemical reactions can't distinguish between stereoisomers. Biology can. If Martian microbes show a preference for one form over the other, that's a signature of life. And most importantly, controls. Every positive result must be verified with a heat-sterilized control. That's how you distinguish chemistry from biology. But there's a deeper question here. If we drowned Martian microbes by adding water, what does that tell us about how we search for life in the universe? We assume life needs what Earth life needs: water, organic molecules, moderate temperatures, an atmosphere. But what if we're wrong? What if life on other worlds has adapted to conditions so different from Earth that our methods of detection are fundamentally flawed? Titan, the largest moon of Saturn, has lakes of liquid methane. Temperatures are minus 180 degrees Celsius. Water is frozen as hard as rock. If life exists there, it wouldn't be based on water. It would be based on methane, or something else entirely. Europa, a moon of Jupiter, has a subsurface ocean beneath a shell of ice. The ocean is kept liquid by tidal heating. It's been isolated from the surface for billions of years. If life exists there, it would be adapted to complete darkness. No sunlight, no photosynthesis, chemosynthesis only, feeding on chemicals released by hydrothermal vents on the ocean floor. Enceladus, a moon of Saturn, has geysers that erupt from cracks in its ice shell. These geysers spew water vapor and organic molecules into space. Cassini flew through the plumes and analyzed them. Complex organics, salts, evidence of hydrothermal activity. If life exists there, we might be able to detect it without ever landing, just by flying through the plumes and collecting samples. But in all of these cases, we're making assumptions about what life needs, about how it behaves, about how we can detect it. And the Viking experience teaches us that our assumptions might be wrong. In 2000, Gilbert Levin gave an interview to CNN. He was asked if he still believed the Viking labeled release experiment detected life on Mars. His answer was simple: yes. Most of his peers disagreed. But Levin never wavered. In 2012, he and his team published a paper analyzing the Viking data using cluster analysis, a statistical method that looks for patterns in complex datasets. Their conclusion was that the data is consistent with extant microbial life on Mars. In 2020, Chris McKay from NASA Ames was interviewed about the Viking results. He said, "We cannot rule out that Gilbert Levin is correct. We cannot rule out that there are dormant life forms in the Martian soil. And if we can't rule it out, we need to take it seriously. Because if astronauts travel to Mars, and if they bring Martian soil back to Earth, And if that soil contains viable microbes, the consequences could be catastrophic. We don't know if Martian microbes would be harmful to humans. We don't know if they would be compatible with Earth's biosphere. We don't know if they would outcompete terrestrial life or integrate with it. We don't know. And that uncertainty means we need to approach Mars with caution, with humility, with the recognition that we might not understand what we're looking for. So here we are, nearly 50 years after Viking, and the question remains unanswered. Did we find life on Mars in 1976? The data says maybe. The scientific consensus says probably not. But the more we learn about Mars, the harder it becomes to dismiss the possibility. We know there's water, hidden in ice, absorbed in salts, possibly flowing as brines in the subsurface. We know there are organics, detected by curiosity in 3-billion-year-old sedimentary rocks, complex molecules, possibly of biological origin. We know there's methane, appearing and disappearing in ways that suggest an active source. We know the environment is harsh, but we also know that life on Earth thrives in conditions we once thought impossible. Boiling acid pools in Yellowstone, frozen lakes beneath Antarctic ice, deep ocean vents where sunlight never reaches, salt rocks in the driest desert on the planet. Life finds a way, so maybe it found a way on Mars too. And maybe, just maybe, we killed it by trying to help it. The Viking labeled release experiment was designed with the best intentions, with the best science available at the time, with the assumption that life requires water, but Mars is not Earth. And if life exists there, it might be so different from what we know that our very presence destroys it. This is the paradox of the search for life. To find it, we have to look. But looking changes what we find. And sometimes, in our attempt to understand the universe, we alter it in ways we never intended. For now, all we can do is learn from Viking, design better experiments, make fewer assumptions, and approach Mars with the humility it deserves. Because if life is there, hiding in the salt rocks, waiting for the next drop of moisture to wake it from cryptobiotic sleep, we owe it to that life not to make the same mistake twice. The next mission to Mars will carry instruments more sophisticated than anything Viking had. They will search for biosignatures, for chiral molecules, for motility, for metabolism, and this time, they will do it without drowning the very life they hope to find. The answer is out there, somewhere beneath the red dust, waiting. We just have to be careful enough to find it without destroying it, and patient enough to accept that some questions take more than one mission to answer. Mars has been waiting for 4.5 billion years. It can wait a little longer